So it's a pleasure to be here to speak to you all with my program director, Natalie Rodriguez. I'm really glad that we're able to co-present because we truly are a team running these programs that we'll tell you about today. So as you heard in the introductions, we have a number of studies, including the International Perinatal Outcomes in the Pandemic Study and the Child Cohort Study. But what we're going to focus on today is our research in and around human milk. So there we go. Starting with disclosures, these are our funding sources and affiliations. And I was asked to share for a moment some wellness tips, which I think is a great tradition to have going here at Rounds. So for me, during this pandemic year, my wellness tip would be spending time outdoors, which is something I actually didn't do that much of before the pandemic, but I've kind of discovered as a way of keeping myself sane. And you can see in that photo there at the top, I have earbuds in. So a lot of times when I'm out and about, I'm listening to podcasts or audiobooks. And so I've shared here a couple, and Nat and I tend to share recommendations with each other, read the same books, listen to many of the same podcasts. So these are some of our favorites. I've got Brene Brown here and Sean Achor, The Happiness Advantage, which are two books that I enjoyed over the past year, and a couple of podcasts. Shiny Epi People is a really fun one where they interview epidemiologists, which doesn't sound that fun off the bat, but they actually have a lot of silliness and talk about everything other than their epidemiology work. And I'll let Nat speak to her photo and a podcast.
Yeah, so thanks very much for the intro and for having us today. I have a pre-existing health condition, and so for myself, it's important that I have supervised physical activity. And so I've actually, I'm at the Refit Center. That's actually my trainer, Evan, who works with me weekly. So physical activity is important, and Refit's done a really good job of protocols and safety, which again, in my case, is important. And as Megan said, we've started a book club. So we read these books and then we break them down and review them. It's been a really nice opportunity for us. And so another book that's maybe not on this list that we read that was quite impactful was Melinda Gates' the views changed, sorry. Melinda Gates' Moment of Lift. And How to Fail is an interesting podcast because it shows there's much to be gained from failure, almost more than from our successes.
Interrupt for a minute. Are we sharing screen?
So I thought so, and then I saw a note in the chat, switch screen share, so I swapped it. So are you seeing the right screen now, Nat?
I've been seeing the correct screen.
Yeah, I think so now. Okay.
Okay. Sorry, good. All right. So those are our wellness tips. And I'm always happy to receive book and podcast recommendations from other people if you want to throw them in the chat or reach out to us later. All right. So on to the main event. Our objectives for today are firstly to explain the dynamic and personalized nature of human milk as a biological system, why we're so interested in studying it. And to really show how Manitoba is pioneering global innovation in this research area. And also, if we have time, to discuss some of the tensions that arise when supporting and studying breastfeeding, because it's certainly a complex area. And then finally, to discuss some opportunities for many of you as clinicians to engage in some of the translational research that we're doing. So I want to start with this, personalized medicine. You saw that in the title of the talk. Many of you are clinicians. So I think this is a bit of a loaded term. It means different things to different people. But I know that until recently, when I would see this term, I would think of things like a personalized dosing regimen for a medication, maybe a personalized cancer therapy that's adjusted and chosen based on the genetics of a tumor, or even more lifestyle things, a personalized exercise regimen. Natalie just mentioned hers at the Refit. But nowadays, when I see this term, I think about human milk. And so this is a quote I love from Cesar Victora, who's an epidemiologist and Gairdner Award winner, from a series in The Lancet on breastfeeding, which if after today you're inspired to learn about breastfeeding and human milk, this would be a great place to start. It's a series of a couple articles really summarizing the state of the art in this field of research from different disciplines. And so he says that human breast milk is not only a perfectly adapted nutritional supply for the infant, but probably the most specific personalized medicine that he or she is likely to receive, given at a time when gene expression is being fine-tuned for life. And you can see the graphic down here. Improving breastfeeding practices to meet the WHO recommendations could save more than 820,000 lives a year. And so that was estimated from some of the extensive meta-analyses that were done as part of this series. And it includes not only child lives, as you would expect, but also maternal lives, because breastfeeding has benefits to maternal health as well. And so I've underlined there fine-tuned for life, because this really encapsulates this one quote, everything that we do in our lab. So we study human milk, and we study not only its nutritional properties, but also the other bioactive functions that milk has. And we focus also, even outside of milk research, on the developmental origins of chronic disease, or DOHAD. And that really refers to the first 1,000 days as a critical period for development. And those 1,000 days are calculated, of course, not from the day you're born, but the day you're conceived. And originally that field of thought started as the fetal origins of disease, really focusing on pregnancy and the importance of exposures during that gestational period for the fetus or child. But it's expanded to look beyond the period of pregnancy into the first 2 years of life, if you were to count those 1,000 days. And I think it's not coincidental that that is the period of time during which breastfeeding is recommended, really emphasizing that this is a critical exposure or experience in the first period of life that has potentially lifelong impacts. So of course, there are many great reasons to breastfeed, and I hope that as pediatricians, many of you are familiar with at least some of these reasons, and we're not going to have time to cover them all, but what I want to focus on and what our research most directly addresses is the health benefits of breastfeeding for the infant. So just a few of those would be some immediate impacts. So breastfed babies have a lower risk of sudden infant death syndrome. They have a significantly lower risk of respiratory tract infections in the first year of life during the time when they're breastfed. Lower risk of type 1 diabetes, so a potential lifelong impact, and a lower risk of obesity later on in childhood and even into adolescence and adulthood. So those are just of the few reasons why we're interested to study breast milk and understand how these benefits are conveyed. But actually, the reason I got interested in breastfeeding was none of those that I just mentioned, and it was all about the microbiome. So this is the cover of CMAJ almost 10 years ago, 9 years ago, and this is a paper that I published during my postdoctoral training. And it was not the first, but one of the early papers describing how the infant microbiome was affected by the way a baby is born, so vaginally or by C-section, and also by how the babies were fed. And so from there, I got really interested in breastfeeding and how it was impacting the microbiome. And fast forwarding 5 years later, this paper was on the cover of Nature. This is not from our group, but from a different cohort called the Teddy study, and was a really comprehensive view at what shapes the microbiome. So to give you a sense of how far the field had moved in 5 years, our study was published on 24 babies, and the Teddy study was several thousand. So the technology enabling us to study the microbiome has really advanced a lot during that period. And this is one graph from that Teddy paper where they look at, whereas our paper looked just at C-section and breastfeeding, their paper with a larger cohort of infants was able to look at many other factors, including maternal body composition. You see here probiotics, vitamin D, and these bars are representing the proportion of the microbiome variability that's explained by each factor. And what's very obvious is that breast milk is, by miles, far and away, the most important factor shaping the infant microbiome. This is in the first few months. It's a longitudinal study, and when they go on and the babies are older, you can see that other factors are relatively more important. But early on, it's definitely the breast milk that is shaping the microbiome. So that was my entry into being interested in breastfeeding. And so really as the way we think of human milk, I mentioned in that first quote, it's really more than just food. And it is really a biological system, and the field is kind of shifting to think of human milk in this way. So it's not just a food, it's not just a bioactive fluid, it's really a system with all these different components. So this is a beautiful piece of artwork from a group called Human Milk, and you can visit them at humanmilk.com. It's a group of artists and scientists that work together on knowledge translation initiatives to help other people understand the complexity of human milk and how important it is. And so you can see here that they've named all the different vitamins and fats, amino acids, and so on that are present in human milk. And I think this is really how we have to start thinking of it in our research and other perceptions. But then it's not just a system in and of itself. Human milk is a system within a system. So it's comprised of many bioactive components, but it's also within this system of the mother-infant milk triad. So often we talk about the mother-infant dyad, but this is a perspective piece published last year in Science by one of my collaborators, Lars Bodie and colleagues. And they really propose that we should be thinking more along these lines, where you have the mother and infant breastfeeding and then the milk itself. And so you have breast milk components, you have the infant gut microbiome, which I mentioned is my particular interest in human milk. And then you have all these other different omics, which either come from the mother or the infant and the communication between them. And that by applying new computational methods and thinking of this really as a triad is really the gateway to understanding the maternal and infant health benefits of breastfeeding, and to even understand if there are new therapeutic concepts we could learn from milk, perhaps apply to other stages of life, health conditions, and to understand the practices we should be recommending around breastfeeding. And if you were to look at the author list in this paper, I've highlighted here, it's a really nice illustration of how interdisciplinary research is critical in breastfeeding. So one of the authors is from the Mother Milk Infant Center of Research Excellence. Another is from a Genomic Sciences and Systems Biology Institute. One is from the WHO, the World Health Organization, and another from a medical school. So I think really we need to be thinking from the clinical, the policy, and the basic science perspective to have the biggest impact in this field of research and many others. So human milk, as I mentioned, is a system. It has many components, and this figure, of course, will be too small for you to read, but it should give you the message that breast milk really contains a lot of things that aren't present in formula, as hard as we might try to replicate. It's also dynamic, which is one of the things that I think a lot of people don't appreciate about human milk, and which is, of course, not replicatable in a formula. So human milk varies diurnally. So from morning to night, the components in milk are changing. Even within one feeding. So the milk at the beginning of a feed is different in composition from the milk at the end of a feed. And this is thought to be one of the ways by which infants learn to self-regulate their appetite and satiety. It varies across lactation. So as the infant is growing and their developmental needs change, the milk composition changes to match. And it definitely varies among mothers. So different mothers are making milk of a different composition. We can think of milk as personalized nutrition. So it is matching the needs of the infant as it goes. And I put here human infant because, of course, all mammals make milk, but it's differing in composition depending on the species. And so if you think just two examples, cow's milk, which is the basis of formula, versus human milk, they're designed essentially for what those infants need. Or those offspring need early in life. And so you can think of a cow, if you've ever seen a calf be born, they're born and they get up almost right away and start wobbling around. And whereas a human infant will spend a lot of the first months of life sleeping. And that's because they're using their energy to grow their brain, whereas a cow is diverting its energy to grow its muscles. And so therefore the milk they're consuming is adapted in that way. So human milk compared to cow's milk, for example, has a really high level of omega-3 fatty acids that are important for brain development. And so you can imagine then if we start feeding milk from different species to different offspring, that's not going to be optimal. And then finally, we can think of milk as personalized medicine. So milk is delivering immunity to an infant when it's born and has a very immature immune system of its own. And not only is it delivering immunity through things like antibodies and other immune factors, but this is optimized to the environment that that mother and baby share. So if the infant is sick, there's a way of communicating that to the mother, and then her body will ramp up production of antibodies to those pathogens. And by the same token, if the mother is sick, then she'll ramp up production of those antibodies to protect the infant from the same infection. So really fascinating in many ways, as you can tell. It intrigues me to no end. So I wanted to try something a little interactive. I know it's difficult on Zoom, but you can throw your answer in the chat and I'll watch them pop up. So I'm wondering, of course, you cannot read this picture, I hope, because it will give away the answer. So taking a temperature check of the audience, can you name something that's found in human milk that is not found in infant formula? So I'm seeing IgA as the first answer, good one. I'm getting a lot of attention lately in the COVID epidemic. There are researchers studying if a mom has COVID, will her milk contain antibodies? I see stem cells, good one, probiotics, antibodies, growth hormones, very good. So these are all excellent answers. and some of the things that we are interested in studying in the milk samples that we have. So this is an image that gives you the same impression, but you should hopefully be able to read. So if we think about formula, it contains all the basic nutrients that an infant would need. Oligosaccharides, lipase, excellent. And so that includes micronutrients, macronutrients, but then on top of that, human milk, breast milk contains many other things. So some of them you caught in the chat: antibodies, hormones, growth factors, enzymes. So these things are not found in formula. And even if we were able to inject them in there, they wouldn't be personalized to that baby and that mother. And that figure is maybe a little bit out of date. It's missing a couple of my favorite items that several of you in the chat were also calling out. So probiotic bacteria are found in milk, prebiotic oligosaccharides, which are food for those bacteria, and we'll talk a bit more about those today. Stem cells, I saw someone mention, which is pretty fascinating. These stem cells actually take root, so they come from mom through her milk and take up residence in the infant's tissues. So brain, liver, pancreas. There are, of course, not many of them compared to the infant's own cells, but they do end up residing there, and it's still kind of a mystery. What is their function? And then also food antigens. So many of you know that we have an active food allergy research program at CRIM. And so I'm interested from that perspective in the food antigens from mom's diet that are showing up in milk and the role they might have in developing tolerance. I see some formulas have probiotics. You are right, and you'll see an updated picture soon. So from here, I want to tell you a bit about our overall research program and then give you one example of a study that we've done. So a lot of the initial work that I did in and continue to do in breastfeeding and breast milk has been within the child cohort study, which hopefully you're familiar with. This is now into its more than 12th year. So this is a pregnancy cohort that started back in 2009 from 4 centers across Canada, Manitoba being one of them. So Eleanor Simons is currently leading the Manitoba site, previously led by Alan Becker. So with a focus on allergy and asthma, but really beyond that, looking at health trajectories in early life and taking a broad view of how genetics and also various environmental factors influence child health. And so the child study is where I had done that original microbiome research and became interested in breast milk and breastfeeding as a postdoc. So when I started up my own program here in Manitoba, some of the first questions I was asking was around breastfeeding or breast milk feeding, and you'll see why I make that caveat in a moment. And to understand just epidemiologically how these different feeding patterns in early life were associated with various health outcomes. And I've highlighted asthma as the example I'll talk about today. And then because the child study had collected milk samples, I knew that we would have the ability to look in more detail at different components of milk to understand if there is a connection between breastfeeding and asthma, for example, what are the components that underlie or drive that association? And then because we've collected a ton of information about the families and the mothers in particular, we could ask questions about the maternal factors that either influence milk composition or perhaps influence a mother's ability or decision to breastfeed or otherwise influence infant health. And then we can go one step further than that and think about the mechanisms within the infant. So I mentioned gut microbiome as the reason I got interested in breastfeeding, and definitely one of the biological systems influenced by breast milk. But there are other possibilities too. So breastfeeding could be influencing epigenetics and gene expression in the infant, could be influencing lung function, immunity, and so on. And then, of course, at the end of the day, our goal with all this research is really to use the knowledge to develop interventions and make a difference in the next generation of mothers and babies, whether that means supporting breastfeeding, optimizing milk composition, if such a thing is possible, or addressing some of those underlying factors. And so a lot of this research is done in human cohort studies, but I also work with, and this is one of the great things about being a member of CRIM, other scientists who do animal models, which are sometimes necessary to address mechanisms, and also with researchers at the Center for Health Policy who have access to population level data in Manitoba. And you'll hear a bit more about that later. So I'm going to give you one example of a study that we did in child around breastfeeding. And so this again is not for you to read, but just to emphasize that in the cohort, we've asked a lot of questions about feeding repeatedly, starting at birth. So we have hospital records from the mothers and infants, so we know what they were fed from the first day in hospital, and then questionnaires every 3 to 6 months out to 2 years and even beyond. And so we've asked a lot more than simply, did you breastfeed your baby, which is sometimes as good as it gets in other cohort studies. We asked questions about how long they breastfed, when they started introducing formula, if they did, what type of formula was it, When did they start introducing foods? If they use bottles, were they glass bottles or plastic bottles? So many layers of information. One question I will draw your attention to is this one. So when breastfeeding since birth, did you use a pump to express the milk? And I had actually not even noticed that this question was asked when I was first doing my research, but it was a PEDS resident who's now on faculty. So Dr. Annika Klopp, when she was doing her residency, had been doing a rotation in the NICU and seeing that many of those vulnerable preterm infants are receiving their mother's milk, but they're receiving it through a tube or maybe eventually in a bottle. And so she asked me, does it matter if the milk is delivered at the breast or in a bottle? And I said, I've never thought of that. And I don't think we could even figure it out because I don't think we asked the moms, but we looked back through the questionnaires and we were excited to find this question. And it wasn't initially asked for the purpose of understanding pumped milk versus feeding at the breast. In the way that we were thinking, it was asked, put there by our environmental scientists who are interested in plastics and contaminants. But nevertheless, the question was asked. So we were able to dig back through and kind of reanalyze some of the data that I had analyzed from the lens of breast milk versus formula and break that breast milk group down further to understand if the method of feeding made a difference. So this was the paper that we published together, and I'll just show you one key figure from it. So here, we've looked at the diagnosis of possible or probable asthma at 3 years of age in the cohort, and we've divided the infants into how they were fed early on. So DBM is direct breast milk, so all breast milk fed at the breast. Formula is formula feeding, and here we're looking at 3 months of age, how are the babies being fed? And so you can see that the formula fed babies had the highest rates of asthma by 3 years of age. The direct breastfed babies had the lowest, but in between that, you have babies who were fed a combination of breast milk and formula. And then very interestingly, you have this group getting indirect breast milk. So they're getting some of their milk pumped and fed from a bottle. And they're intermediate, and the effect estimates up at the top here are after we adjust for important confounders like the mother's own asthma, whether or not she's returned to work and sent the child to daycare, which might be one reason for feeding pumped milk, and so on. But we still see these significant differences. So the conclusion of this paper was that compared to direct breastfeeding, any other mode of infant feeding was associated with an increased risk of possible or probable asthma by 3 years of age. And I'll tell you, and Annika could confirm, we reanalyzed these data many different ways because it surprised us at the time. There wasn't a lot of literature on the differences between pumped feeding and direct breastfeeding, a couple of papers, but this was quite a robust finding, regardless of how we analyzed the data or the different confounders we considered. So when we thought about why that might be, some of the ideas that occurred to us, which we're still investigating, it might be the bioactivity of the milk. So once a mom has pumped and kept the milk in the fridge or perhaps frozen it, maybe some of those bioactive components, including the probiotics or the live bacteria, might be inactivated. There might be differences in the physical lung exercise, and there actually is one study of a person who has measured the lung force needed to suck from a bottle versus at the breast. It might be about the skin-to-skin contact, which could, again, relate back to the microbiome or other things. Temperature regulation occurs through skin-to-skin contact, and that may be missing if baby's being fed by a bottle from somebody else. Also, this infant mother signaling, which I alluded to earlier. So the idea that if babies got an infection, then that can be communicated back to mom through components in the baby's saliva during breastfeeding. So there really needs to be that direct contact, which doesn't happen with bottle feeding. Or it could be that there are contaminants from bottles or pumps, which I mentioned was the original reason for asking that question. So many possibilities that we're still investigating. And I want to make my usual disclaimer. Unfortunately, this is sometimes how our research has been portrayed in the media. So pumped breast milk contains nasty bacteria. It is harmful and causing asthma. This really isn't the message that we have intended to convey. And really, I think what our research shows is that pumped milk is not equivalent to nursing at the breast, but it does suggest it is beneficial compared to formula, which is a good thing. And we know from other research that pumping can absolutely help sustain breastfeeding. So this might be the only way for a mother to provide her own milk to her baby if she's got to be somewhere else, if there are problems with latching and so on. But at the end of the day, it's important to find out why. And so as initially a basic scientist, this is my curiosity, is there is a difference. Now let's find out why, because if it's about the way babies are being fed from a bottle, that can be changed. If it's about the way the milk is stored, that can also be changed. So at the end of the day, understanding why will allow us to help all moms and babies, including those who are pumping. All right, so moving on to the milk itself, and I promised you an updated version of that photo. So this one you'll see does have probiotics and prebiotics occurring in some formulas. And so as we have learned more about the microbiome, some formulas are starting to add more and more components to become closer and closer to human milk, and probiotics and prebiotics are among those. So in the milk collected from the child study, which is nearly 3,000 mother-infant pairs, I've been able to analyze several components of this rainbow of human milk, always in collaboration with other experts. And so the one that I'll give you an example of today is human milk oligosaccharides, done in collaboration with Lars Bodie at UC San Diego. And we have several other studies underway and an ever-growing wish list. So as our one example today, I want to tell you about human milk oligosaccharides. So HMOs are the third most abundant solid component of human milk. So milk is primarily water, but if you look at those solid components, the most abundant is lactose, followed by lipids, and then outnumbering even proteins are human milk oligosaccharides. And so there are many different types and configurations of these oligosaccharides. They are non-digestible carbohydrates, meaning that they're not digested by the infant. However, we now know they are digested by the infant's microbes. They are structurally very diverse. So all mammals make oligosaccharides, but they make different combinations of them. So for example, cows make about 40 different oligosaccharides in their milk, whereas humans make over 100. So to me, this is automatically a reason to be fascinated by oligosaccharides because they're one of those species-specific components of milk. They're highly variable. So it's not that all human mothers are making the same 100 oligosaccharides. They're actually making different combinations. And we actually don't know very much about why that variability occurs and how it might be important for the infant. And then finally, we're still learning about the impact of HMOs on infant immunity and the microbiome. It's really since the microbiome became discovered as such an important component of our health in the last couple of decades that we've become, therefore, interested in these HMOs that are present in human milk. So this is one example of data from our research in the child cohort study. And here we've profiled over 400 mothers. And you can see from the mom who's making the least amount of HMOs to the mom who's making the most amount, there's a really big range. So almost a fourfold variation in the amount of HMOs in this milk. And if you look at the individual, so here we've measured the top 20 HMOs in different colors, you can see that if with individual variation, there's up to 100-fold variation in individual HMOs. So huge variation. What you can see as one important factor is I've divided the moms here into non-secretors and secretors. This refers to one gene, actually a one base pair polymorphism in the FUT2 gene. And so if you have one configuration, you're a secretor and you make this blue oligosaccharide, which is called 2-fucosyl-lactose. And if you're a non-secretor, you don't make any of that 2-FL. But it's not only 2-FL that's affected. Many of the other HMOs are present in different amounts depending on this single base pair polymorphism. Of course, within those two groups, there's still a lot of variation that we're working to understand. But one thing that I would draw your attention to is this is the one oligosaccharide 2-FL that is starting to be added to infant formulas. And so in the sort of ever-going pursuit of making formula more closer to human milk, this is one of the components that's now being added. And you can see that, as I mentioned, about a quarter of mothers don't make any of this 2-fucosyl-lactose. So I think it's interesting to consider whether it's a good thing to provide it to every infant when some of them would have never seen this oligosaccharide in their mother's milk. So what difference does this make to the baby? I think this is the ultimate question that we want to understand. And so one example of this is that we looked at food sensitization in the child cohort. So here we've got 59 babies who became sensitized to foods by a year of age versus others who didn't. And we wanted to understand whether the milk they were consuming had different levels of HMOs and whether they might be important in programming this element of health. And you probably can't see those individual graphs too well, but that's okay because the bottom line is there were no associations. So none of these 19 different HMOs were different in the babies who remained healthy versus became sensitized. But when we looked at the overall profile of these 19 HMOs, we did find a very significant difference. And so to me, this message says there's no single HMO that is the silver bullet that's going to predict or prevent food sensitization, but the overall profile does seem to be quite important. which makes sense because after all, babies are receiving all of these HMOs, not one by one. Another way to look at these data that you might be more familiar with is a receiving operating curve. So you can see here the AUC perfect would be 1.0, useless would be 0.5. So this analysis gave a 0.73 AUC, which is considered good. But remember, this is only looking at HMOs. There are so many other components of milk. And of course, we're interested in other conditions. So we're moving forward with our research program to understand if we look at more than just HMOs, can we get even better at this prediction and understanding the system of milk and relate it to other health outcomes. So those are a couple examples of the research we're doing in milk and in infant feeding, and it leaves a lot of open questions. So what really is a healthy milk composition? I think there's probably no simple answer to that. It's going to depend on context, and it's going to involve not only the nutrients, but also the bioactives. How do breastfeeding or lactation or human milk impact both maternal and infant health? We've still got a lot to learn there in terms of the mechanisms. And what are the individual, population, and systems level determinants and consequences of the vast variability in human milk? So I think looking at the individual moms, their individual genes and polymorphisms is one thing, but also having to think at the population and systems level, because we know that there are many population level factors and policies that affect whether or not moms are able to breastfeed. And we don't want to lose sight of that. Also, as I've mentioned, it's important, I think, to look at human milk as a whole with all these many components, but that is a computational challenge for sure. Once we understand this, is it possible to optimize human milk composition? Are there supplements or other things we could advise mothers to do to improve their milk composition? And taking that maybe one step removed, are there things we could do to optimize donor milk provision for premature infants? Thinking even beyond that, could we harness what we learned from human milk for applications beyond infancy? So there are interesting research projects going on about using HMOs, for example, to treat autoimmune disease in adulthood. And so at the end of the day, I think it's important that we learn how to collaborate across disciplines to answer these questions and accelerate progress. And so here's where I'm going to turn it over to Natalie to speak about two of our main initiatives in Manitoba, which I think are really leading the way in terms of human milk research. So the Manitoba Interdisciplinary Lactation Center. This is a photo from the day it was launched and announced with the Minister of Science and the CFI that provided the funding, and then the International Milk Composition Consortium funded by the Gates Foundation. And I'm pretty proud that we made the front page of the Free Press the day this was announced. And Natalie and I were invited to go out and visit the Gates Foundation shortly thereafter and had a really interesting time. So Nat, over to you.
Great, thanks so much. So really a springboard for our international research is the Manitoba Interdisciplinary Lactation Center. And this was funded by the Canadian Foundation for Innovation, which Megan mentioned previously. And, you know, it really is about innovation and discovery. And that's kind of, you know, a basis for a lot of our research. It's unique in that it captures infant feeding information, and this is opportunistic. But it captures administrative data securely. We will then take the raw milk data. We have milk samples within our biorepository, which is state of the art, and we're linking. So this really brings in that element of personalized medicine. So not just the raw milk values, but what's going on in a mother's life, you know, and what's going on at visits, anthropometrics for the infant, and gives us a better look at milk. Megan, next slide. So you can see for the infant feeding database, it's opportunistic data collection. And I think for clinicians, this is important. Having worked on the clinical side of things myself, I know anytime we introduce a research project, the question is staff and burden. All of clinics are busy, and there just really isn't the ability for, you know, clerks, et cetera, nurses, to conduct additional questionnaires. This is quite interesting in that it takes information from routine vaccination visits, it links it with Manitoba Health Insurance Registry, and it actually populates electronically into a form. We do have some really amazing rural practitioners who actually populate forms by hand, and then it's actually scanned, so either electronically or the physical paper form, and routes to a virtual secure research environment where we can then de-identify the data and link it. And so this provides opportunities for a whole host of future research initiatives. So social and structural determinants of breastfeeding, interventions to support breastfeeding, and pharmacotherapies and breastfeeding. Another interesting piece of this is we are looking at additional questionnaires, and the funding from CFI has enabled us to have a milk coordinator who can go into the clinic and actually consent mothers and ask additional follow-up questions. And we're even going so far as video for consent and explaining our studies to make it, more accessible to mothers. Often research programs and studies can be overwhelming, and we really do want to break down those barriers. Next slide. So a big part, or the hub, of our GATES project and our other projects is the Human Milk Biorepository, and that resides within CRIM. And I have to say, CRIM has been amazing. Much of this biorepository was built during the beginning of the COVID pandemic. So we have -80 smart freezers. Those actually link to your phone or your tablet. They have touchscreen capability. They will create an airlock. You can see when the freezer's been opened, how many times it's been opened. They're hardwired, but they also have battery backup. And those were en route when the COVID pandemic started. And so much of our machinery and equipment has been, put in place during the COVID pandemic. It also features the Unity SpectraStar near infrared analyzer. And we use that to analyze macronutrients within the milk. And we do have matrix cryotubes, which are cryotubes that have a 2D barcoding system. So there's actually a QR code on the tube itself, as well as a digital barcode, and it goes into a rack, which has its own barcode, and we can scan entire racks of breast milk. This becomes important when you're dealing with high sample volumes, and also for liquid handling. So these racks fit perfectly into liquid handling systems in our partner labs at UCSD, at Cedars-Sinai. And so you can see Kelsey Fair and Larissa Latosky there at work in the biorepository itself. And I have to say, Kelsey has been absolutely amazing in receiving samples, processing these samples. And part of this is that we are using a laboratory information management system. So you can see that there's 1,200 human milk samples processed. 1,200 seems like quite a bit on the surface. If you are dealing in this case with 400 samples from a field site, those are actually sub-aliquoted into, further for 5 partner labs. So 400 samples becomes 2,000 samples. And then those are shipped all over the world. So it's important for us to know where each sample is, be able to track them, see what's taken out of the system. And, I have to say, Kelsey just does an amazing job. We have, even cryo labels. So they are on foil and they're actually laser printed, and, she's doing all of that work. Next slide. Part of our work as well is clinical. So we have done work with Michael Narby, and that is one of our postdoctoral fellows, Dr. Shreyas Kumbhar, and they're looking at human milk versus bovine milk fortifiers and the impacts on gut microbiota and oxidative stress. So there's work ongoing in the NICU there. And Marilee Brockway, another one of our postdoctoral fellows, and Dr. Jeanette Festival run they're working on the MOM study. So again, that's looking at the secretor status that Meghan had mentioned, but they're also our partners at the Northern Star Mother's Milk Bank. There is actually a donor drop here in Manitoba, a satellite of Northern Star. It's also important in that Northern Star Mother's Milk Bank actually provided a milk pool, which is the quality control for our IMiC study, which I will talk about a little bit later. And that is funded by the Bill and Melinda Gates Foundation. And that's allowed us to not only provide quality control samples for all of our analyses, but is allowing us to compare geographically diverse milk pools. So we'll have the Canadian pool versus milk pools from low and middle income countries. So you can see for the IMiC study, this is really groundbreaking in that, as Meghan said previously, we've only had snapshots of milk and not really been able to look at it comprehensively. And IMiC really has enabled us to look at milk composition in its entirety. And that's unheard of. So we have 5 field sites for the IMiC study. We are looking at Canada, Burkina Faso, Tanzania, Nepal, and Pakistan. And again, personalized medicine. So this is where, impact, who is this impacting? And that's one thing that with Gates is incredible. So previously, it has been too expensive to look at milk composition as a whole. The infrastructure just was not there. And you wonder, who does it impact? So in Pakistan, for example, 60% of infants, there's evidence of stunting at 6 months. And when we talk about the first 1,000 days, we're finding in low middle income environments that there's stunting in growth at even 2 months of age. In Burkina Faso, for the Masame study, our partners there describe situations in which formula is not available for complicated births or preterm babies. And so if wet nurses in the community are not available, those infants actually do not survive. So we're looking at infant mortality rates. So again, this really is an opportunity for impactful research. And from an analysis perspective, we are looking at macronutrients with our colleagues at USDA, micronutrients as well with our colleagues at USDA, bioactives, microbiota. So we're doing targeted and untargeted metabolomics. We are looking at all sorts really of analyses, whether it's proteomics. We're even involved in the Global Health Vaccine Accelerator platform with Gates, in which pathogens are printed onto microchips and exposed to breast milk. Again, coming back to even those early days, we know that milk changes. And so we're looking at early milk and colostrum and seeing those effects on pathogens. So this is a really impactful study. And then we're taking that personalization to an even further layer. And we're pairing it with questionnaire data, so metadata, anthropometrics, to give us a more fulsome view. And we actually have this amazing opportunity to work with data partners and machine learning experts, both at Berkeley with Dr. Alan Hubbard and Stanford with Dr. Nima Agiapour, and they're actually doing predictive modeling, so harmonization across these cohorts and predictive modeling on infant growth outcomes. And this previously has not been done. And I have to say, a lot of the analyses we're doing have not previously been done in breast milk. So, we've undergone validation studies, and we are shipping samples as far as Austria for analyses in these cases. And it becomes interesting to say, how do you do that? How do you get samples from all over the world, receive them in Manitoba, process them, and then ship them across the globe? And breast milk is temperature sensitive, so we need to keep it at -80. So it's using couriers that can replenish that dry ice along the way. For some of our stakeholders, for example, in Burkina Faso, there is no reliable source of electricity, in which case we've had to use rechargeable breast pumps that can be taken into the field. And so you really learn with low middle income, stakeholders, or kind of anytime you're taking it into that discovery space, you just have to be creative in your thinking. And I think that our lab really does that brilliantly, and this team does it brilliantly. And it's just amazing, to be a part of this study. So knowledge translation, again, why does this matter? How do we get our information out there? And we really do embrace a team approach and a team science approach to everything we do. Too much of research is done in silos. And if we aren't helping people and doing impactful research, then we're not doing this right. We're getting it wrong. And so for us, it's very, very important that our findings, are disseminated to the public. So you'll see we've had quite a few features now, whether it's in journals, being featured on the news. Megan's done several talks for CTV. I've had presentations for CBC. And so it's not just basic science, it's the clinical side of things, it's knowledge translation to the public. And we need to be doing research that matters to our stakeholders. And so I always say we don't do research on our stakeholders, we research with them. And that's very important because they're helping to inform our research. Next slide.
So I want to also point out.
Go ahead, yes, the conversation, yeah.
Yeah, so we have, in addition to the news features, been involved in developing courses about breast milk that are aimed at clinicians. We've done some videos that are public facing. These come out of the child study to educate people about breast milk. I've been part of a committee convened by the National Academies of Science, Engineering, and Medicine in the US, which was convened by various government agencies to do a really deep dive on the evidence of human milk nutrient composition to inform nutrient guidelines. And then also 2 of our trainees, as well as several other members of the lab, have been involved in writing articles for the conversation. And then Nathan and I, so Nathan being the co-director of Milk, have several positions that help us translate knowledge into other societies and communities. So Nathan is the scientific chair of breastfeeding on the American Public Health Association. I'm a member of the Begin working group that's been convened by the NIH to develop new funding initiatives for human milk research. Nathan is the research and evaluation lead for the Baby Friendly Initiative of Canada, a quality improvement project. And I have several roles on the International Society for Research in Human Milk and Lactation. So I think together, these different initiatives help us really make sure the research we're doing gets out into practice and policy. So with that, in our last couple of minutes, I want to briefly discuss tensions in breastfeeding research and give you some ideas of how to get engaged. So probably, as you're listening to me talk and Natalie talk, you may have been thinking to yourself, but not every mom can breastfeed, or but is there really evidence? Are we guilting moms? And so this happens a lot in the breastfeeding field. And when I think about it conceptually, I kind of think about these 2 sides. I guess if you're not making somebody upset, you're not doing anything really impactful. But these are some of the messages we often get. So on the one hand, we have people who are so committed to breastfeeding they can't ever envision a reason why any baby should receive formula. They really believe that every woman can breastfeed if she just tries hard enough. We know that's not true. And to them, formula kills babies, period. And like any research that could even potentially help formula companies is evil, because it obviously is there to be helping formula companies. So we hear this on the one hand. But then on the other hand, there's also a fraction of people who really deny that breastfeeding is useful at all. And so to them, there's really actually not any hard evidence that breastfeeding is beneficial. We know that's not true. To them, promoting breastfeeding is anti-feminist because women shouldn't have to breastfeed. To them, fed is best, and this is sort of a whole movement, and actually breastfeeding promotion kills babies, because if you're forcing mothers to breastfeed and their babies are not getting enough milk, then that could be harmful to the baby, and some babies even could die. And to them, breastfeeding research is evil because really it's just guilting mothers who can't breastfeed or didn't want to, and that is harmful. So you have these extreme views, and then I feel like there's us in the middle, and really we're doing research on human milk and breastfeeding promotion to provide evidence to inform science, practice, and policy that ultimately should help all mothers and babies regardless of how they're being fed. But sometimes this is how it feels to be in the middle, getting heat from both sides. It can be difficult. And so what I've come to think about is that there is really a spectrum. So there are some people on one side that really believe everyone should breastfeed all the time. And there are people on the other side that believe nobody should ever have to breastfeed. But in the middle of that, there's a lot of reasonable people with different perspectives, and they won't always agree for sure, but they can have productive debates and discussions. And so what we've tried to do as an approach is to ignore those really extreme views because it's not productive to engage. And among the rest, find ways to collaborate across disciplines towards the shared goal, because I think really at the end of the day, we all want to support maternal and child health, and then you can feel a little bit happy. So one example of this is that 2 years ago, we convened a workshop with breastfeeding experts from around the world. None of you probably know breastfeeding like we do, but these are some pretty big rock stars in the field who we were able to invite to come to Winnipeg in February, no less, in a polar vortex. And we had a really great workshop of conversations. And this does include people from, the Center for Economics, Center for Health Policy, Center for Social Sciences, in addition to people looking at infectious diseases, inflammation, neonatology. So it was truly interdisciplinary, and we had some really good discussions that resulted in this publication that was just published last fall that you'd be welcome to check out. In addition to publishing the paper, we also did a piece for the conversation. And so in this workshop, we really addressed those tensions and conflicts that occur within the field. And then we recently organized some public outreach events to reach beyond just the clinical and scientific community. So to finish off, how can you become involved? This is a photo of some of our milk members at a Café Scientifique that we held last year. We do have a milk club that meets monthly. It includes researchers and trainees, community health workers, clinicians and policymakers. So if you're ever interested in joining, please reach out. And then there are other things you could do. So Natalie highlighted 2 of our clinical trials. We're always interested in collaborating if you have ideas or want to become involved. We, of course, are going to need help recruiting research participants. The MILK biorepository hasn't launched in a big way yet because of COVID, but when it does, we'll be looking to recruit moms from clinics to provide milk for research. Also collecting data, building partnerships is really important. So we want to continue to expand our partnerships with the clinical community. And then finally, sharing evidence. So once we generate this research, we know it needs to get beyond other scientists, and you can be of help in that way for sure. So we have a website where we've compiled a lot of our favorite resources. So videos and articles and infographics. So if you're looking for resources, that's a good place to start to pass on to your patients or colleagues. So I want to end off thanking everyone in my lab who has done a great job of contributing to this research or supporting the team in other ways. A special shout out to the child cohort study, the local and national staff and researchers involved, and to our colleagues at MILK. So with that, we will end off and be happy to take any questions. And if you want to reach out some other time, here's where to find us.